Hi everyone. For my protein project, I will be talking about the COL1A1 protein, which is the building block for building type 1 collagen, or the most abundant type of collagen within the human body. The COL1A1 protein is found within connective tissue systems within the human body. So this includes bone cells, skin tissue, muscles, tendons, as well as vasculature cells. Specifically, I'm going to talk a little bit about a mutation in the COL1A1 protein and how this can lead to decreased protein function as well as the development of a rare bone disorder called Caffey's disease. Caffey disease is characterized by excessive bone formation or hyperostosis. I'm hopeful that after watching this video, you also will have a better understanding of Caffey disease and its molecular mechanism. Type 1 collagen can be characterized as a heterotrimeric molecule, which is composed of 2 alpha-1 chains of collagen and 1 alpha-2 chain. The COL1A1 protein is the building block for synthesizing the alpha-1 chain of collagen. Together, these 3 chains weave together to form the signature rope-like structure which is associated with collagen. By packing tightly together to form a dense scaffolded matrix, molecules of type 1 collagen offer physical structure to the cell, and then at the organismal level, they offer support and tensile strength. In order to understand the molecular basis of Caffey disease, I am going to use clay models which I made to display the primary, secondary, tertiary, and quaternary levels of structure for the COL1A1 protein. Let's get started. The primary structure of the COL1A1 protein is repeats of the glycine XY triplet, where X and Y are oftentimes proline. On the top, you can see that I have an amino acid sequence which corresponds to the wild type COL1A1 protein, whereas the sequence on the bottom codes for a mutated COL1A1 protein. As you can see, the difference between a healthy patient and a person suffering from Caffey disease is a single amino acid substitution at position 836 of the primary COL1A1 protein structure. Whereas a healthy individual would have arginine, a large amino acid with a polar positively charged side chain, a person with Caffey disease would have a cysteine, a smaller amino acid with a polar uncharged side chain. When we look at the secondary level of the COL1A1 protein structure in just a second, we'll see how this substitution leads to structural differences in the wild type versus mutated COL1A1 protein. The secondary structure of the COL1A1 protein is a left-handed helical coil, as shown below in the model. The secondary structure is stabilized by hydrogen bonds formed between the carbonyl oxygen of one amino acid and the amide hydrogen of another amino acid within the primary structure. Because the secondary level of protein structure is determined by the hydrogen bond interactions between the backbone of the primary sequence, we can forget about side chain interactions until we reach the tertiary and quaternary structure. As you can see, the secondary structure of the COL1A1 protein is a uniform left-handed coil, and it remains constant regardless of whether or not a patient has Caffey disease, because arginine and cysteine differ only at their side chains, which are considered only once reaching the tertiary level of protein structure. The tertiary structure of the COL1A1 protein refers to its overall 3D orientation in space, and is primarily determined by interactions between the side chains of amino acids within the polypeptide. As you can see, the tertiary structure is a triple helix composed of 2 alpha-1 chains, shown in blue, and 1 alpha-2 chain, shown in red. For a patient with wild-type COL1A1 protein, the coil structure is uniform in both size and shape, whereas the structure is completely inconsistent in the case of a patient who has a mutated COL1A1 protein. There are several reasons for this. First, cysteine is a unique amino acid because it is the only amino acid which can form disulfide bonds, or covalent linkages, which are stronger than other forces contributing to the tertiary structure of COL1A1. Disulfide bonds are sort of like molecular safety pins, keeping parts of the 2 alpha-1 chains firmly attached to one another, which helps explain the discrepancy in coil size and shape in the case of Caffey disease. Another factor to consider when discussing tertiary structure is the electrostatic interactions between side chains. Both arginine and cysteine have polar side chains which can participate in dipole-dipole interactions as well as hydrogen bonding, but they have different charges. While arginine's side chain is positively charged and interacts favorably with negatively charged side chains from other amino acids, cysteine's side chain is uncharged. So, when arginine is replaced with cysteine, electrostatic interactions within the protein are completely altered, potentially causing inconsistencies with the diameter of the coil. Finally, the quaternary structure of protein refers to the interactions between multiple different polypeptide chains. In the case of type 1 collagen, multiple triple helices come together to form a tight staggered matrix, which is known collectively as a fibril. As was the case in tertiary structure, disulfide bonds and electrostatic interactions are involved in the formation of the quaternary structure, ultimately resulting in the inability for consistent, organized collagen fibrils to form in a patient with Caffey disease. The replacement of arginine with cysteine at position 836 of the COL1A1 gene affects the protein structure by creating variability in collagen fibril size and shape due to the presence of abnormal disulfide bonded dimers. At the level of the cell, the extracellular matrix of connective tissue cells in patients with Caffey disease is less able to offer mechanical support and tensile strength, which are 2 functions that are crucial in an organism's well-being. At the level of the organism, this mutation causes an abnormal and rapid bone growth, swelling of soft tissue, fever, and pain. Finally, I'm going to conclude by discussing 3 limitations of using clay for my model. First, the use of clay meant that I had to ignore the flexible and resilient nature of collagen. Baked clay is rigid and brittle, which makes it an inaccurate representation of collagen's unique elasticity. Secondly, we talked about how thousands of molecular interactions are involved in the formation of collagen structure, but my clay model simply cannot accurately represent this complex chemical interaction. Finally, my model doesn't allow for the detailed portrayal of collagen's eloquent higher-order structure, which is often involved in the context of the human body. Using clay doesn't allow for a detailed depiction of this complexity, and ultimately serves as an oversimplification of how collagen functions within the human body. Thanks so much for watching, and I hope you learned a little bit more about Caffey disease.